The purpose of this experiment is to use and initiate online biological exploration tools for the human genome. We will use these tools to research the FXN gene and see if there is a relationship between it and pancreatic cancer. With this experiment, we can understand how a researcher can come to identify cross-biological knowledge available in databanks online. Let's first go to the Ensembl website. Select our species as human and search for the FXN gene. Follow the link for the gene drop-down menu and notice the number of transcript variations available. Let's go back a page quick. And now look at the location of the gene. Note this location. Because we'll use it to compare locations for pancreatic cancer research. We're going to export this data just so we can get a feel for it. We can input this data into other tools. Export as HTML and FASTA, and you can notice the code here. We're now going to do the same thing for pancreatic cancer. Again, go to the gene section and notice the number of variations in the parentheses. We're going to scroll down till we find one with a similar gene locus, chromosome 9 specifically. And that would be the CDKN2A gene. View the location again and note its similarity. Probably not significantly close. Export the data again as FASTA format and HTML. If we wanted, we could save and export this code and use it in other tools. Let's now go to the ncbi website and do a similar search. Type in FXN. We'll go down to the gene section and click on it. Look at the number of results. There's quite a few. Let's just go with the first one. Look at all the information displayed, location, size. You can find the sequence. We're going to scroll down till we see the ncbi reference sequence so we can access the mRNA sequence. It's about 2/3 of the way down here. Click on it. Now we are going to view it in FASTA format, like we did earlier in Ensembl. And we can send this code, this sequence, if we'd like, which we're going to do. Complete as a file in FASTA format, and we will create that file, and it'll be in our download section for later, in our download file. and we can use it in a tool later. Let's do the same thing for the pancreatic cancer gene. This time we'll just search straight CDKN2A. Again, we're trying to distinguish the relationship here between FXN and pancreatic cancer. Click on one. View it in FASTA format again. Notice the sequence here. We'll save it as a file. FASTA format again for later. Now let's do a similar search again on the EBI website. First, FXN. And we will select the human from Caxton. Notice the data listed, our location, everything. We will go to the protein tab on the left. And we will select View Sequence in UniProt. We can click on the different isoforms if we'd like. We can also view this in FASTA and use a number of tools from this location. But let's go back quick. And we'll select the isoform 1 and use the BLAST tool to find more genes and more sequences of a similar structure. This will run it right in the EBI website, and our results will be sequences with a similar format. The brightest green will be our closest matching sequence. Notice the sequence similarity bar there. Red is less similarity. We're going to select a few and then align them so we can match them up. So select 5 or 6 and click Align on the bottom right. This will line up our sequences and we can compare them. Again, running this tool directly in EBI. Once that alignment is done, we're going to go up in the top right corner and click Gel View. This will allow us to view this sequence in another way. Click Keep and open up the file. So launch Gel View, and Gel View is useful for a number of different things. Definitely visualization techniques is a great one. Agree and click Run. Close out of those little ones, we'll keep the graph. So this shows us a number of different comparative similarity techniques here on the bottom, showing our alignment on the top. The nice thing about JavaView is that it's kind of a home-based usage, so we can import files that we've saved on our own computer if we'd like. And we're going to go ahead and do that. Click File and from File, and we should go to the Download section and open one of the 2 sequences that we saved previously, and that'll add that right in there. Again, we're going to do it with our other sequence, the second sequence from CDK, that gene. And now we have all of our sequences in there, and we can compare them. So we'll use a visualization technique here, the phylogenetic tree, to compare the distances apart, percentages apart. Calculate tree, and you can just select the first option. And here we have our tree showing the similarity and relationships between the other sequences. Now we can exit out of these, and we're going to do one more location, one more tool-based server, and it's called BioExtract Server. This is probably one of my favorite. If you haven't done so, register by clicking the register section. Take a minute here. That way we can save workflows. I'll show you how to do that. I'm just going to sign in quick. Once you've signed in, go to the Workflows tab, and on the left at the top, create and import workflows. And this will allow us to save workflows for later usage. You can share them as a group. So yeah, just click record workflow. And now everything that you do from here on until you hit save will be recorded. Go to the query tab, and we're going to search FXN again. Excuse me. Select ncbi proteins. There we go. Now select gene and FXN. And then we're going to add one more search line to narrow it down a little bit more. And in this one, define species and human. And then let's search our query. And for this BioExtract server, we'll pull them all up right in the extracts page, and we can use these files directly from this page without having to save all the data. So here's our query. We're going to pick a couple of these. We're going to narrow it down. You could view the record and have all the details about the gene, similar to EBI, ncbi, ensembl. Select, and we will check box the one that we would like to keep, and we will keep only selected records. Narrow it down a little bit more. And we could export this file if we'd like. We could save it on our own drive, use it in GelView if we didn't like using the tools from a BioExtract server, but personally I do. That's right here. Now we will go to the Tools tab and use this query in some of our tools. It'll take a little bit to load. There's quite a few tools available through BioExtract Server. Go to Similarity Search Tools and BLASTP. We'll make sure that extracts in FASTA format is selected, and then we're going to go down and we're going to select only from the Swiss protein data bank. And then you can execute the tool again, and our results will show up in the extracts page this time. So now we can just head over to the extracts page and view our results. So these will be sequences similar to the FXN human frataxin that we selected earlier. So we're going to narrow them down. These are a few species, as noted on the right-hand side of the description, and keep only selected. And now we can go align these sequences if we like. We'll go back to the tool section. It'll take a minute to load. Once it's done loading, now we will go to the alignment tools and select Clustal W. Again, use from extracts page, FASTA format. Make sure that protein is selected there, and execute your tool. And this will line up our sequences that we've just found using BLASTP. Here we go. There it is, sequence alignment. And now I'll show you how that works. Go to the create and import workflows, and now we'll name our workflow and save it, and then in the future we can use these or share these with groups on the groups tab there if you'd like. We can also run this workflow and it'll obtain all the results that we had previously retained, but you won't have to go back and do each step and open each tab. It'll do it automatically. You can obtain all the information you obtained before, which is very nice. So we can click it. It's on the left hand side there. And there you have it. There's BioExtract Server.